Hey guys, today we're going to do human development. And this stuff is fascinating to me that all of this, most of the time, goes right and results in a normal human baby. Um, just all the steps and processes it has to go through. And then even before that point, before fertilization, all the things that have to happen correctly is just amazing to me. So anyway, hopefully this is interesting to you guys. It is to me. And we will jump into our question of the day. Why do most birth defects occur within the first 8 weeks? I do want to go over the pre-lecture with you, so as always, if you feel confident in this, you can skip past this part. Just some definitions here. The union of the sperm and the egg is called fertilization. And once fertilization occurs, this forms a zygote. This must be one of my old pens. There we go. The first cell of the new individual. The head of a sperm is capped with an acrosome. So if I were to draw one of those out here, here's where all the mitochondria are. Here's the flagella. It's kind of like a cap that covers the head of the sperm. And it's an organelle that contains digestive enzymes that are going to help a sperm enter through the plasma membrane of the egg. So it's going to help that sperm get into the egg. Now to the egg, the plasma membrane of the egg is surrounded by an extracellular matrix called the zona pellucida. And that zona pellucida is surrounded by a few layers of cells called the corona radiata. So if we were looking at a portion of the egg here, this would be the zona pellucida. And then you would have a few layers of cells around here surrounding the egg. And that would be the corona radiata. CRZP. Those aren't proper abbreviations, that's just what I'm doing for the picture. So during fertilization, several sperm penetrate the corona radiata. So several sperm will get through this first layer here. And I was watching another lecture on this and I should have written down the numbers. I always say that. But per ejaculation, it's something like, I might be way off. It's a lot of sperm, like millions. I want to say 100 million, but that seems a little high. 153 million? I can't remember. A lot of sperm. And only about less than 100 will actually make it to the egg. And then about 7 will be able to penetrate the corona radiata. So, if that doesn't make you feel special, like you were a fast swimmer at one point, half of you, it should. Okay, after a sperm head binds tightly to the zona pellucida, so a little sperm gets in here and binds to the zona pellucida, the acrosome will release those digestive enzymes that forge a pathway for the sperm. At this point, the plasma membranes of the egg and the sperm fuse, and the nucleus of the sperm enters the egg. So now we have both sets of DNA. inside the egg. And to prevent polyspermy, poly means many. So you only want one sperm getting in there. You only want one half set of DNA. When a sperm comes in contact with the egg's plasma membrane becomes depolarized, preventing the binding of any other sperm. So this is going to essentially, you shall not pass. We already got one in. We're not going to let anyone else in. Okay, the 4 stages of human development with their description. The first stage is cleavage. And this is when there is mitotic cell division. The cells divide, but the whole does not increase in size. So let's say this is your zygote here, your fertilized egg, and it's going to divide, but not increase in size. Those 2 cells are going to divide. Those 2 and those 2 will divide. So we haven't increased in size at all, but the cells have divided. Next is growth. And this is when there is cell division and the cells also increase in size. Next one is morphogenesis. This is when the cells migrate, they start to move where they need to be, and the embryo assumes shape. And last of all, we get differentiation. Which is just what it sounds like. The cells start to become different. They take on specific structures and functions. Just like cardiac cells are going to be different from nerve cells. All right, so on to the lecture here. So pre-embryonic development. During the first week of development, we technically call it a zygote. And so it's going to undergo that cleavage division like we just saw in the pre-lecture where there is mitosis with no increase in size. So here fertilization occurs. Now we have a zygote. And you can see it's dividing in size, or in number, sorry. So this one cell divides into 2, those 2 divide into 4, those 4 divide into 8, but it's still basically the same size here. Then we get to what we call a blastocyst, which you believe is right here. It's calling it morula. I think that's the same thing, or does that come up somewhere else? So a blastocyst is our blastocyst is over here. There we go. Is a hollow ball of cells. With an inner cell mass like you see here. And that inner cell mass is the embryo. So it's what is going to become the baby. So these are the cells that will make the baby, and these will end up becoming the embryonic or fetal part of the placenta. Which brings us to this. It's the chorion. fetal side of the placenta. So if you look at this picture down here, zoomed in a little closer, a little further along in development, this is the chorion here, where the layer where all of the vessels are. So extra embryonic membranes, so extra means outside of the embryo, and the chorion, it's kind of repetitive here, is the fetal side of the placenta. And it's going to wrap around the embryo in a sac. So if I were to kind of outline the chorion here, it has these vessels that extend towards the maternal placenta, the maternal blood vessels. So that would be our chorion. The allantois on this picture is right here. And this is where the vessels emerge from the embryo. You can see them coming out right there. And this will become the umbilical cord. And there also is a yolk sac early on. So if you think about a chicken, I believe the yolk sac provides food for the chicken. That's not what it does for a human embryo. It actually is where blood cells are made until the bone marrow develops and they can be made there. So blood cells made here until bone marrow develops. The amnion in this picture is here. It's the layer that's closest to the embryo, membrane closest to the embryo. And its job is to cushion and protect the embryo. And when you hear someone talking about their water breaking, it's the fluid in that sac that is coming out. It's not a very fun experience, but we'll write that down. Amniotic fluid. When water breaks, this is what comes out. And we forgot to talk about where do identical twins come from? Our question up there. So we call it an embryo from 2 to 8 weeks of development, and it's referred to as a fetus. I hate saying it, but I don't know if it's a boy or a girl. So it from 9 weeks on until birth. So what happens with identical twins is during cleavage, and I don't know why, it splits. So kind of, let's see. let's see, I think it would still be a zygote at that point. During cleavage, the zygote splits. So let's say you've got at like a 4-cell stage here. For some reason, that's going to split. Those are terrible circles, I'm sorry. Split into 2. So keep in mind, this has the same DNA, same egg, same sperm here. So those will end up going on to develop 2 separate babies with identical DNA. Fraternal twins are different. I feel like we talked about this before. Maybe this was at my other job. But with fraternal twins, there are 2 separate eggs fertilized by 2 separate sperm, and therefore they're no more closely related than just regular siblings. They just happen to be born at the same time. But with this, it's the 1 egg, 1 sperm where all this DNA came from, and so if it splits super early on in development like that, you can get identical twins. One more thing to note down here. So here are the maternal blood vessels. And that is that in the placenta, the fetal and maternal vessels meet. And so this is the site of nutrient and gas exchange. So obviously this baby can't eat or breathe on its own, and so that's where it gets its nutrients and oxygen. It gets rid of its waste and CO2. But it's important to note the blood does not mix. It only exchanges. Sorry, getting crowded here. So the only time when the maternal and fetal blood mixes is during childbirth, and that's why we get problems with like the positive and negative blood types. So it's not harmful during the pregnancy, it's when the mother gets exposed to some of that fetal blood during birth. I don't know why it mixes. I would imagine something to do with all of this coming apart. But it would be on the subsequent pregnancy that there would be issues. So. But that blood does not mix. All right, implantation is next. This usually happens in the beginning of the 2nd week. So the zygote will come and the chorion invades the uterine tissue and grows into the endometrium, so it kind of implants into the uterine wall, and blood vessels are made that extend out into the endometrium. And with something called an ectopic pregnancy, this is when the embryo implants somewhere that is not the uterus. Somewhere it's not supposed to. So implantation outside the uterus. And if that's the case, they're not getting those blood vessels extended to them that they need, and the embryo can't get nutrients and oxygen, and so the pregnancy is usually terminated because there's just no way that baby can grow and develop and get the nutrients and oxygen that it needs. It's also not safe for the mother, typically, depending on where it implants. Okay, HCG, human chorionic gonadotropin. This is secreted by the chorion around week 3. And keep in mind at this point, the individual probably doesn't even know they're pregnant yet. And this is what is detected by pregnancy tests. The good old stick that you pee on. And what it does is it acts as a luteinizing hormone, which will maintain the luteum, which maintains the pregnancy because it blocks FSH, follicle stimulating hormone, and luteinizing hormone, and so it maintains the endometrium. You don't want it sloughing off because you have a baby developing there. Gastrulation is what happens next. So this is around week 3. And this is when we get to morphogenesis that we talked about in the pre-lecture. So stuff is going to start to migrate and move around. Cells migrate to create layers. And the embryo takes shape. And we're going to form what we call the primary germ layers, which is what this diagram over here is showing. The endoderm, the mesoderm, and the ectoderm. So the endoderm, endo means within or inside, and so that's going to be like your digestive tract. Mesoderm, I see that M and think muscle. So this will end up developing into your muscles, your bones, your circulatory system. Circulatory system. And then the ectoderm. Ecto again is outside of. So this is going to be like outside of your body, and this develops into your skin and nervous system. So it's kind of color coded over here. We have our ectoderm here in blue. So this will end up developing into the skin and nervous system. We have the mesoderm, the middle layer here in red. It's going to end up becoming the muscles, your circulatory system, your bones. And then the endoderm, the inner layer, which will end up, it's in yellow here, being your digestive tract. So I would know those 3 and what they end up forming. We need to describe this picture right here. So this is hard to see. Even on my printed copy, I can barely tell what it says. But villi from the chorion which are these little spikes that are kind of sticking off that you can see. So villi from chorion. Digest some of the endometrium. And then the endometrium grows back and surrounds the entire sac. You see here. Remember, that was during implantation up here. Sorry, rewinding a little bit there. All right, continued embryonic development. This is 8 weeks on. Those germ layers, endoderm, ectoderm, mesoderm, give rise to cells which differentiate into specific cell types. So this is where we have differentiation occurring. And the organs develop and as the body form takes shape. So by 8 weeks, we've already differentiated into specific cell types. The organs are developing, the body form is taking shape, which brings us to our question of the day. Why do most birth defects occur in the first 8 weeks? This is because when most of the body and organ systems are developing, and after that, it's mostly just growth. And the thing is, a lot of women don't know they're even pregnant within these first 8 weeks, so they might not be taking care of themselves the way they need to. Maybe smoking, drinking, not taking vitamins, and that's when all of these developing organ systems are taking shape. So that is crucial. So if you are of childbearing age and there is any possibility you could become pregnant, I would take a prenatal vitamin. Plus, it will make your hair nice and your nails grow faster. That's non-professional advice, by the way. Do what you want. Okay, so our answer to the question is most body organ systems develop by then. After that, it's mostly growth. Obviously, there are things that can go wrong after 8 weeks, but kind of at this point, what's done is done, and then it's just growing after that. So let's talk a little bit more about the placenta. This secretes both estrogen and progesterone during pregnancy, which will inhibit FSH and LH, follicle stimulating hormone and luteinizing hormone. So this is going to inhibit follicle development and ovulation, which is good. We don't need any more eggs here. We have already got a baby growing. And progesterone maintains the endometrium to support the pregnancy. So why don't women ovulate when they're pregnant? Because that placenta is secreting that estrogen and progesterone, excuse me, which will inhibit FSH and LH. So we don't have follicles developing, no ovulation, no eggs getting released, can still be sexually active and not have another baby. And then the progesterone part helps to maintain the endometrium. Embryonic blood vessels infiltrate the endometrium. Sounds like a war. And gas exchange occurs between blood vessels of the mom and baby, but again, that blood never mixes. So if you look at this picture here, we've got the chorionic villi that have infiltrated here. And then you have the maternal blood vessels. They don't mix, but they are close enough that they can exchange nutrients and gases there. The umbilical cord contains vessels that supply blood to the embryo, and you have umbilical arteries and umbilical veins. And it's kind of backwards of what you would think. The umbilical arteries carry deoxygenated blood. From fetal tissues to the placenta. So in our model here we have the placenta. This is where gas exchange occurs and the blood gets oxygenated. And the umbilical arteries are going to be carrying deoxygenated blood from the fetal tissues back to where it can get oxygenated. The umbilical veins are going to carry oxygenated blood, because it's picked up oxygen here from the placenta, from the placenta to back to the fetus so it can be distributed to the fetal tissues. So for the exam, I would know that the umbilical arteries carry deoxygenated blood and where it's coming from and where it's going to. And then that the veins are carrying oxygenated blood and where it's going from and coming from and going to. So like probably be able to draw this out and understand what's going on here. Right, last page. Look at that cute little developing baby. So with fetal circulation, most blood is going to bypass the lungs. The baby's not breathing, so that's not where oxygen exchange occurs. But there is still some blood needing to go there because those lungs are developing and they're going to need things like oxygen and nutrients. So let's note up here that most blood must bypass lungs. Lungs are immature and not needing air. But still, some blood goes there to grow lungs. I feel like this is the messiest packet I've ever done. I'm sorry. Okay, with fetal circulation, there are two things you need to know about. The foramen ovale and down here, the ductus arteriosus. so let's start with the foramen ovale in our picture. It's right here. And it's a hole between the right and left atria. So some oxygenated blood entering the right atrium is diverted to the left atrium. With the ductus arteriosus is located here in our diagram. And this connects the pulmonary artery to the aorta. So again, most fetal blood bypasses the lungs. So for these two, for the exam, I would know this bullet point here, and then know what it's bypassing, like with the foramen ovale, and this is typical flow of blood. You're bypassing from the right atrium to the left atrium and skipping all of these things that would lead to the lungs. And with the ductus arteriosus, you're going from the pulmonary artery to the aorta and skipping all of these parts. So know this. Not necessarily the names of all these things, but this is just the lungs in here, bypassing the lungs. Okay, at birth, both of these bypasses close or should close. So the foramen ovale, just the inflation of the lungs on that first breath of air, creates a vacuum that pulls blood from the right atrium to the lungs. And this forces the flap to close over the foramen ovale and seals. Just a side note, if you've recently taken anatomy, you might think I sound like an idiot. For some reason, I learned it as foramen ovale, but I've heard people say foramen ovale, which is probably correct, but it's just in my brain to say it that way. So, I mean, it is what it is. Okay, ductus arteriosus. At birth, cells will grow and close this duct. And so that will end up becoming what sounds like a Harry Potter spell, the ligamentum arteriosum, are those cells that grow and close that duct. So with preterm infants, they're usually on a respirator because they haven't made that pulmonary surfactant yet, and so their lungs tend to collapse on themselves. So they use a respirator. To help draw blood to lungs. And both bypasses, because of that pressure, usually seal on their own. But if they don't, it may require surgery. Okay, that's that's it for human development. We've got 2 packets left after this one, and they're both on evolution, which is, I think I say this about everything, my favorite concept in biology. So get ready for that. I hope to have that up soon, and we'll see you then.